Hi guys, welcome back to my YouTube channel. So if you've forgotten to subscribe, please make sure you've subscribed and hit on the notification bell. Today we are going to discuss on introduction to microscopy, the types of light microscopy. Thank you, hope you'll enjoy. There are two major types of microscope, the light microscope and the electron microscope. So today we are going to deal on the light microscope. The types that we are going to discuss on today are the first one, the brightfield microscopy, the second one, the phase contrast microscopy, and the third one, the differential interference contrast microscopy. So under the first one, which is the brightfield microscopy, there is a unique characteristic in it, which is the contrast of the microscope is brought about by staining the specimen. The advantage of this is that it's easy to use this brightfield microscope. It doesn't need huge adjustments, so the adjustments that you need are few, so easier to use. And then the disadvantage of it is, as you stain your specimen, you're going to add some extra details that they are not being needed. So that's why the brightfield microscopy is not used common. Type of light microscope is the phase contrast microscopy. Under this, the source of light is always directed to the condenser lens, and then the condenser lens directs the source of light to the specimen. At the specimen of course will have two types of regions, the thick and thin regions. So as the light penetrates through the thick and the thin region, you'll find that there will be light interference. And since the phase contrast microscopy from the name has a phase plate, the phase plate will be able to convert the light interfered to form altered brightness, and this will be an advantage to the microscope because one will be able to detect the living specimen and the tissue culture. The disadvantage of this type of microscope is that one cannot be able to detect a thick organism. The third type of light microscope and the last one is the DCI microscope, which stands for the differential interference contrast microscopy. Under this, you'll find that the source of light is narrated to the polarizer. Then it passes through the polarizer to the Wollaston prism. At the Wollaston prism, that is where the light is split into two beams. And then when the specimen is present there at your stage, the beam of light is going to recombine, and the recombination will be different depending on the thickness and the thinness of the specimen. And so as the light recombine, there is going to be the light interference, and this aspect of light interference is the one that is going to help you study the living system. So the disadvantage of this is that there is, due to that enhancement of light and overshadowing of the specimen, there's going to be a lot of distortion on your image. Guys, please subscribe. If you've not subscribed, subscribe.
Hi guys, hope you're good. Hope Mazim Melan. I'm feeling so much entertained. Yeah, so I think mesoma pia kama mimi. So subscribe to the YouTube channel and make sure you click the notification bell to get notified of the next video. Yeah.
So guys, you've all learned. So make sure that you've commented down below and suggest on any topic you want me to discuss on. Thank you.